just turn off the mic before you say.
Yeah, it's all.
It's pretty slow.
Hello. A lot of progress has been made. They came up with
Still like I'm using my computer.
So, when you
No. Don't want to leave.
yeah,
What is that?
when you like change
Are you
it from
excited?
Like fully virtual.
You going to first?
Probably food factory. Excellent.
To Barcelona.
Yay.
Yay.
Should I stop and track?
so like just put it back.
Also doing photos.
That's
what?
I'd love to have words.
Oh, last week was the first time I didn't come to seminar.
If I didn't have my own.
Yeah.
I just skipped this.
I didn't know that should appear. So you're gonna have
What happened at what?
Today I didn't want to come.
Yeah.
Girl, no one wanted to come.
What is this?
Does anyone ever want to come?
Transfer refrigeration. Let's says we should start.
So how was your morning? Yeah, I saw your pics. So cute. Where'd you go? The restaurant looked nice.
I've never seen it.
Okay, hello everybody.
Very nice.
Welcome to today's seminar. Welcome to the BME seminar series hosted by the Department of Biomedical Engineering here at Columbia University. My name is Josh.
Yeah, the umbrella.
Davis. Of today's meeting. We are continuing to offer this lecture in a hybrid format. Thanks, guys. Our speaker today will present for about 40 or 45 minutes, and we'll leave the last 15 minutes or so at the end for question and answer. For our virtual audience, throughout the presentation, please present your questions using the Q&A feature at the bottom of the screen rather than a chat. The Q&A feature lets us better keep track of everything and organize. We will try to answer as many of your questions as possible during the Q&A session at the end of the meeting. For our live audience, of course, we'll have an interactive question and answer session that I'll be hosting, and I'll come to you with this microphone as always. Today, we are excited to welcome Professor Jude Phillip. Dr. Phillip is an assistant professor of biomedical engineering at Johns Hopkins University. He has secondary appointments in the chemical and biomolecular engineering department, oncology, and he's a core member of the Institute for Nanobiotechnology as well. He completed his Bachelor of Engineering in Chemical Engineering at CCNY, a PhD in Chemical and Biomolecular Engineering at Johns Hopkins, and he did postdoctoral training at Weill Cornell Medicine. At Hopkins, his lab studies biological aging dynamics in health and disease, and he combines fundamental engineering approaches with aging and oncology research to develop translatable strategies and technologies to probe aging and disease. So welcome, Dr. Phillip. Screen sharing set up here. How does that look, Alexis? Good?
Sounds good.
Thank you so much, Josh, for the kind introduction. And I'm really excited to be here today to share some of our work with you, the audience, and in terms of the title, Aging at Cell Resolution. So the title is a bit, something we're working towards, but I think we've been making a lot of really cool strides in terms of bringing this to fruition. So my lab really studies, we lie at the intersection of engineering and medicine, and we focus on biological aging dynamics. So some of the key questions that we try to answer is related to how some of the changes that occur as we age really inform our overall health and disease, as well as understanding the microenvironment of lymphoma. But today I'll mainly focus on aging in the context of health and disease. So the talk will be structured in the sense that I'll frame the problem in the sense of asking some of the key questions. Why is it important to study aging from my perspective? And why do we age differently? Then I'll tell 3 small stories in the sense of a way that we're using to develop dynamic cell-based biomarkers of aging. I'll talk a little bit about a tool that we recently developed to be able to profile cells, dynamic properties of cells. And then one of the projects that we're just recently getting some really nice data about in terms of like mapping senescent cells. So it comes as no surprise, I think, to most of us that the aging population within the US is increasing, right? And as a matter of fact, based on US Census data, there's a projection by the time we get to 2050, about 1 in 5 people would be above the age of 65, right? But the interesting thing about that is while it's great that people are living longer, this length of time living, this longevity doesn't always translate in terms of living healthier in the context of health span, right? So what this is projected to do in terms of the socioeconomics and overall health is to shift the balance in terms of the health of individuals away from being healthy and towards a state which is characterized by multimorbidities, so the same individual having multiple different diseases. And as a result, that kind of like gets to the point of polypharmacy, where individuals have to take many different types of medication. And that really, looking at that through the lens of the health infrastructure that we have in the US, really blows up the healthcare cost, right? But I think, we can all appreciate as engineers in the room is in the sense that although it sounds like, these challenges, using engineering innovation and engineering approaches, we should be able to transform those into unique opportunities, right? That we could pretty much push this forward. So over the last couple of decades, really the field of aging has been focused on trying to understand and map some of the changes that are occurring with age, right? And a few years ago, there was this paper published on the hallmarks of aging. So what that tried to outline is these 8 different features of individuals, many of them focused at the molecular scale, so understanding molecular type changes in terms of how people change as the passage of real time, right? And at the same time, not only changes don't only happen at the molecular scale or at the physiological scale that we can see or physicians can actually measure, but it also happens at the cell scale where cells change their properties, right? Their ability to deform or their ability to translate forces and so forth. So in the sense, there's a lot of these changes that are occurring across multiple different length scales. So in my lab, when we think of aging, we think of it through the lens of scales such that every change that we can observe at the physiological level is associated with changes both at the cell level and the molecular level, and also it interacts at the upper scale in terms of epidemiology, right? So we can break aging into these 4 scales, molecular, cellular, these macro phenotypes or clinical, as you can think of it, and then these epidemiological scales where they're all connected in time and space, right? And they're connected in time and space, but the exact connections in terms of how one scale maps to the other is not well understood, right? So we're at this interface where we're trying to understand how some of these molecular changes map onto cell changes, map onto clinical changes, as well as connecting some epidemiology. And to do this, we develop new technologies. We do mechanistic-based research to understand biological discoveries with the whole goal of implementing this into the clinic to actually impact people. And I think looking at it at this scale also gives an added perspective, is that it allows you to really look at aging in a multi-dimensional sense, right? That we can not only look at for instance, molecular determinants that really drive aspects of our aging. But from the other side, we can really look at, for instance, how things like disparities or pollution and environmental changes really impact that. So today I'll focus only on the cell-based level, right? Which we think of as this nice bridge that really connects the molecular world to the outside world in the sense that it funnels through and changes functional phenotypes of cells. So in my lab, we try to get from characterize these living systems. We work primarily in human samples and animal models, where we use a variety of different tools and approaches in deep phenotyping to profile cells, and then having information about individuals at the different scales, we can use systems biology-based approaches to really combine and to use these approaches to stratify in efforts for precision health and precision aging. So to start this off, one of the questions I think about a lot is in the sense of why do we age differently, right? And one of the ways I like to really illustrate this is that think about you're interacting with three groups of people, right? All of them are aged 80 years old, but in one group, they can run marathons. In another group, they're all bedridden. And in the last group, they're somewhere in between, right? So what we really see is that there's this inherent variability among individuals as we age, right? And this really brings us to the realization that there's potentially a nonlinear association between our chronological age and our biological age. And biological age is a term that's been defined in the field to try to capture the risk of disease that really comes as a function of the passage of time. But many times we've also heard the saying or the line that age is the biggest risk factor for disease X, and you can fill in sort of like that disease. But then we can also think about what is the real risk factor? Is it age? Is it chronological age? Or is it biological age? And how do we use this notion of chronological age and biological age to really inform what I call convergent phenotypes that really allow us to map changes that occur in individuals? They may not all occur at the same age or at the same rates, but if we can identify these convergent phenotypes, we may be able to map these nonlinear associations, which is connected to this multidimensional way that is each person is defined, right? And this is just a simplification in the sense that you could think of it as an interactome where each person is seated at the center of molecular changes, cellular changes, clinical changes, and epidemiological changes, and they all interact, right? So in terms of identifying targets and changing or modifying our aging trajectories, we can really develop new approaches to do this. So the goal is kind of like to think of each of us as we age, we're going along this trajectory. And if we can map your trajectory and understand what are some of the features that inform that trajectory, can we modify and change upwards? So when we started looking at this, we started by just asking, what can our cells tell us about our age, right? And to do this, we took a cohort of patient cells, donor cells, that was all part of a study in Baltimore called the Baltimore Longitudinal Study on Aging, right? So these are groups of all healthy individuals. So they have no comorbidities, right? So they're healthy and they're taken from individuals across the age range, right? And here I focused on individuals ages 2, which is very young, to age 96, right? So kind of like that, looking at aging not as discrete populations, but as a continuum, right? And then we took these cells and from these individuals, and they were dermal fibroblasts taken from the arm, and then we exposed them to this slew of different mechanical measurements, right? And we measured both molecular changes, right? And on the right side, you can see we did 5 different types of measurements that were associated with this hallmarks of aging, right? So looking at things like cytoskeletal organization, how cells respond to stress, the secretions of proteins as well as energetics. And then on the other side, we focused on some biophysical measurements that tries to capture the functional states of cells, right? So how do cells move, both at the single cell level or collectively, or how do cells exert forces on their environments? How do cells deform, or the viscoelasticity or the plasticity of the cells? And also something that pathologists look at a lot in terms of their morphology, right? If I put them on a surface, or I put them in a three-dimensional gel, what morphological states do they assume? So from all these different classes of measurements, we came up with about 208 different features that really describe the cells and the cell behaviors of these individuals. And we took the averages and then just plotted them on this correlation plot where we can look at the associations with age, and this is chronological age in healthy people, across these multiple different measurements, right? So ATP, we can look at secretions, we can look at in a biomolecular sense, but also morphology and motility and some of these other force mapping measurements from the biophysical sense. And then what we were able to see is that there were some very strong associations between the age of the individual and some of the features, right? So for instance, we see this band up here in orange that showed that there were quite a bit of parameters from the morphology space that centered around the variability within the population, which is kind of like the CV of these different measurements that associate strongly with age. As well as if you look at the blue dots all the way at the bottom, you see there's strong associations with motility, which is kind of like how these cells are moving in this two-dimensional space. But one of the things we realized is that if we were to use this to actually predict the age of individuals, just using a purely univariate regression model, right? What we see, and I'm not showing this data here, but the error of our prediction blew up very quickly, right? So our approach to sort of like, to correct for this was to combine parameters together, right? So we did sort of like, we use a generalized linear model, but now using a bivariate prediction, right? And this is just showing a surface of how combinations of these different features really inform the correlation and eventually the prediction of some of the age in these healthy individuals. And one of the things I'll leave you with for this part of the story is in the sense that we wanted to find what is the cheapest way that we can measure multiple of these that is able to predict the age pretty accurately across multiple individuals. So we took about 40 different individuals, and then we predicted their age, and we plotted it against their chronological age based on purely morphological features, right? So things like nuclear size and the variability of the protrusions in terms of the cells. And then what we saw is that in healthy individuals, we're able to see this good correlation. And then if we add in some samples of accelerated aging conditions, like Hutchinson-Gilford progeria syndrome and Werner syndrome, you see that it sort of like breaks the correlation. But I think it shows it so much more clearly if we calculate this age differential and plot this age differential, which is just the difference between our predicted age and our chronological age, and map that against the chronological age, and we're able to see that individuals fall into these three kind of like stratas, right? Where we can call it expected aging, where the band is very narrow, kind of like, nominally in terms of aging. But then you also see some individuals that have accelerated aging, and most of that group were the actual samples from people with accelerated aging conditions like progeria. And then we also saw some remnants of some individuals that seem to maybe aging at a slower rate, right? They kind of have like these delayed aging. So what this result sort of pointed to us is that aging information is in fact encoded within our cells, right? We can measure things about our cells. There's a certain type of memory that we can take them completely out of the physiological context and still capture the aging behaviors, right? But one of the key questions that we didn't know is how is that information propagated, right? It's there, but how do we propagate it across multiple populations? So one of the things, the approaches that we use to sort of like get at that question is in the sense of, can we use dynamics of cells? In the sense of, we would put cells, whether in three-dimensional gels or 2D substrates, and then measure and track their movement over time. So what this movie is showing you here is actual cells moving within a three-dimensional collagen gel, right? And then from this actual movement of cells, we can track the XY positions in time and then be able to map out their movement behaviors, right? And we can calculate, many of the similar parameters that you could even use to calculate things on a car, right? The speed of the car, or like how far they travel. We could do all that for cells. So in the context of aging, we asked, what are some of these features that are changing with age? And if you look at it at a bulk, in terms of just, bulk displacement, which I'm showing here as a mean square displacement, or the probability density function for cellular displacements and things like that. What we're seeing is that there's a shift with age, right? Cells seem to be moving less. They seem to be changing the direction more frequently, as we can see from the changes in the autocorrelation function and the velocity magnitude. And it just seems like overall, and if you look at D, they're just decreasing in terms of their motility as you increase age, right? But one of the key things that we noticed here is that all of the cells that we're tracking, they're being tracked at the single cell level, right? So we have single cell information about every one of these cells as a function of condition. But when we report the data or we analyze the data, we just take averages and we say average speed, average displacements, and so forth. So that led us to think that we're leaving a lot on the table. So the question is, can we develop a tool where we can capture and take advantage of the single cell nature of the data and then still be able to generate sort of the insights that we would get at the bulk, the biological simplicity, but also get additional information potentially related to heterogeneity and cell state transitions and so forth. So we went ahead and we looked at a subset of individuals ranging in age from 2 again, and this time to 92, and just mapped out all the different cells that we track across all these conditions, right? So the insets that I'm showing here are individual cells taken from different individuals, and they're all centered at 0, right? So we in silico just center them so that we can see the whole pattern of their movement all at once. And then just looking at this, you can see that the footprint or the radial distance that they're traveling is decreasing as a function of age, right? And that fits with what I showed you before in terms of the lower displacement. But if you now just randomly select 25 cells in each of these different pools, you see that there's a zoo of phenotypes, right? There's heterogeneity in terms of the phenotypes. And for instance, within a 3-year-old, you can see cells that move far distances and cells that don't move much at all. And then you can go to an 85-year-old and you can see similar types of trends, right? Cells that move a lot and cells that don't move much at all. So we hypothesized that, this shift that we're seeing in terms of cell movements as a function of age is not due to all the cells shifting their phenotype, but likely due to a fractional redistribution across multiple states, right? So the question now becomes, how can I define these states, right? So we went ahead and did, borrowed from our friends in the single cell world in terms of, single cell RNA sequencing and so forth. We used some of the similar types of approaches and applied them to some of these biophysical features of cells. So we did clustering analysis, and this heat map here is just showing we use 8 different parameters that describe the movement patterns of cells. And then every row is a single cell. And then the colors represent the magnitudes, the normalized magnitude of each of these parameters. So what we saw is that within these, 800 cells or so across the 12 different ages, they fall into these 8 different clusters, right? These 8 different groups that are defined by different patterns, right? So mathematically, they're defined differently, but are they functionally different in terms of like a qualitative way? So what we did was actually take the cells within each of these different clusters and then plotted the actual trajectories of cells in each of these 8 different states. And interestingly, what we were able to see is that you're able to see homogeneous, distinct groups of different patterns in terms of how these cells move, right? So we look at, for instance, P3, you see these cells move very directedly and they move a lot and they're very persistent, right? Whereas if you look at P8, you see that these cells are largely non-motile, right? And it seems to have one main bifurcation where you get 4 and then 4 that are mainly separated based on two factors, right? How their spatial persistence and the distance that they're traveling. So then using these, we plotted it in Tisney space, right? And then now colored each of the dots. So each dot is a single cell. We colored it based on the different clusters that we generated based on the unsupervised hierarchical clustering. And what we were able to see, yes, that they were clustered within proximity of each other within a particular group, right? And then now if we now color the ages onto these, one of the things we also saw, which was interesting, was that each cluster wasn't defined by a particular age, right? But the ages were more or less distributed across these 8 different clusters. So our next question became, can we go back and ask what is the representation of each age across each of these different clusters so that it'll help us to understand these aging trends, right? So here I'm showing you just like plots of these 3 different age groups. So we separated now these 12 people into different age groups based on literature cutoffs, right? So how the body of work in the literature really like sets these thresholds. So we call it young post-adolescent adults and older adults, which we count as 65 and above. And one of the interesting things we see is that the heterogeneity again defines these trends in age, right? Where we see almost a maximization of entropy, which we use to denote the heterogeneity, and that minimizes as we get above 65 in terms of these older adults. So from the take-home conclusion that we have, kind of like from these first 2 parts, is that although aging information is in fact encoded within properties of cells, biophysical properties of cells, the propagation of some of this aging information seems to be associated with cell-to-cell variations and cellular heterogeneity. So our next question was, because we're understanding such interesting pieces in this aging space, can we generalize this approach, right? Can we develop a comprehensive framework that we can use to understand ensemble motility patterns such that deviations from general trends could inform us about disease? And particularly, we're very interested in understanding age-associated diseases, right? So the next couple minutes, I'll spend some time just like talking about this tool that we just developed, that we're getting ready to submit very soon, that we call HALO, right? So HALO is this computational framework that captures spatiotemporal patterns of single cells, right? So we're capturing both their spatial behaviors, but developing ways that we can also capture the temporal nature of the cells. So how these cells are moving in time. So what this approach does is it takes sort of like imaging, right? So we image cells moving, whether in 2 dimensions or 3 dimensions, in vivo or in vitro. Really, the mode of the imaging doesn't necessarily matter, but then we're able to capture these coordinates, right? So whether it's XYT or XYZT, and then we're able to track these cells, homogenize or harmonize the data structures such that we can combine data sets, and then do feature extractions and then classifications using some machine learning approaches. So one of the first challenges we really had to solve was this notion that the field of studying cell motility is not standardized at all, right? In the sense that in my lab, for instance, we may take images every 2 minutes for 10 hours, whereas in somebody else's lab, they may take images every 10 minutes for 24 hours, right? So how can we effectively combine some of these data sets to be able to get sort of like insights on it, right? It's kind of like that QC step that, to kind of like pool the data together. So we did this kind of like, little in silico experiment where we took the trajectory of a cell, and that's what I'm showing you here, where we had images taken every 2 minutes for 10 hours, right? And then we in silico degraded the time resolution, right? So essentially we're skipping points in between. So we go from 2 minutes and 4 minutes and 6 minutes just to understand how the trends are changing. And then we computed what we call the concordance, which is a comparison to its what we call the ground truth, which is its most time resolved data that we have, right? So here it's 2 minutes. And what we see is that as we start degrading the time resolution, we get to a point where the phenotypes that we're calculating become completely different than at the low time scales, right? So think of it like this, in the sense of if I'm moving around, like meandering through this room, and you're recording my position every 2 minutes, right? And I just, stay here and I just like go around this table for a little bit and then walk over there. If I'm doing that on the time scale of 2 minutes and I'm not able to capture it on a time scale at 4 minutes, you can see how that error kind of propagates, right? And these become particularly important, especially in immune cells, right? Because these immune cells tend to move very quickly and navigate and surveil a lot. So the way that we solve this, and I'm not going to go into too much of the details, but I'm happy to take any questions on it if anybody's interested, is that we developed a way to predict the position of cells at intervening time points, right? And what that involves is a combination of interpolation and an angular deviation, a correction factor that we can predict this state. And then when we compute this concordance, we can see that it's not statistically different to the ground truth, right? Which is what we're trying to achieve. Whereas if you look at interpolation alone, we're just kind of like, finding that scaled midpoint, we see that there's a big difference. And then the second piece is that because now we had these multiple data sources of data, the question becomes, what is the best time point that I should shift all my data to be able to compare them effectively? And what that result showed is 5 minutes, right? So we had data with intervals from 2 minutes all the way up to 12 minutes. And we found if we shift everything to 5 minutes, it's most similar to its ground truth, right? So we're able to capture that pieces of information. Then we went through a similar process to what I explained before. We took, we computed these 8 different parameters for every single cell and was able to do sort of clustering analysis. In this case, we used k-means clustering analysis. And then we decided on the number of clusters based on looking at the stability of the silhouette and also the inertial value, right? So here I'm just showing you the ratio of the inertia on the silhouette. And then we kind of picked at the point where 17 clusters to define the 17 different patterns within these motility states. And just as a quick sanity check, this heat map here is really showing this characteristic staircase plot of the distances from the centroid. So we computed the distance of every cell from the centroid of every cluster. And then along the diagonal, we see mostly blue, showing that the distances are very low, meaning our clustering is the amount of clusters that we decided was good. So we can again plot that into t-SNE space. We see that the different cells clustered within proximity. But in addition to that, these 17 motility patterns broke into these 4 modules that I call these modules that are defined based on these 8 different motility parameters, right? So the t-SNE in the bottom, this red and gray, is showing you how the magnitude are distributed, right? So for instance, if we look at SG4, we can see that it's very red in terms of the mean square displacements. It's very red in terms of the diffusivities and very red in terms of the spatial persistences, right? So if I actually plot the trajectories of those, I expect those to be cells that move a lot and are very persistent, right? So we went ahead, plotted these 17s, and that's exactly what we see, right? In the sense that you're able to identify these distinct patterns or behaviors of cells that we can then use to sort of characterize different disease states and so forth. We also showed that we can build this into a predictive model, that we can predict the classifications of cells that are not part of our data set. So we did that two different ways. We did a 90-10 split in a training set. So we have about 75,000 cells, split it 90%, 10%, trained on the 90%, and then predicted on the 10%. And then we can get about a 99% accuracy in terms of our classification of the 10%, right? And then similarly, we simulated trajectories. So we simulated 75,000 trajectories and then predicted the classification for each. And then the way that we identified the true class of these predicted ones is to iteratively add in one cell. So we did it 75,000 times, one cell into a trained model of the other 75,000 from the training set, right? So that way we're not shifting significantly. We're still able to retain the rigidity and now able to get the true class of these. So how can we use this, right? So an example that we do, so we've been doing a lot of immune work now, right? In the sense of trying to map aging in immune cells, right? So we took cells from 3 different donors, right? So this plot that I'm showing you here is the representation or the abundance of cells characterized within each of these 17 motility patterns, I mean. And then each individual, right? Donor 1, donor 2, donor 3. And these are all at baseline. And what we see is that there's distinct patterns that define each person at baseline, right? However, there's a certain number of, certain groups that seem to be characteristic of monocyte populations, right? So that's kind of like the one you see towards the middle, those like higher abundances there. So we asked, if we took those monocytes and we exposed them to pro-inflammatory factors that are present within the cancer microenvironment, right? How do these cells respond? And are we able to really capture the motility behaviors of them? And what we see is that even though each donor has a different baseline, under that perturbation, they respond very similarly, right? In the sense that there's this enrichment in terms of these specific motility patterns. And that is also characterized by a decrease in the heterogeneity. So if we calculate the Shannon entropy, we see that at baseline, the Shannon entropies are always higher than when they're exposed, right? So in the sense that exposed cells or monocytes exposed to pro-inflammatory cytokines are more similar to each other than even to their own baseline, right? Which you can kind of see from the heat map. But then the key question becomes, okay, we're seeing these shifts. Classically, we would just compute like the speed or the displacement or so forth. Can we still tell, the behaviors of these cells in terms of the magnitude, the biological insights on it, in addition to some of the heterogeneity? So if we look at the populations that are highly abundant at baseline and highly abundant at the exposed condition that I'm highlighting here in blue and red, you see that you're going from states that don't move much to states that move a lot, right? So if I had to guess, I would say that upon exposure to these conditioned media, these pro-inflammatory cytokines, I would increase all my parameters, right? Speed would increase, diffusivity should increase, and so forth. And that's exactly what we see, right? You can do it at the bulk, and you show these shifts and increases in the population. So from this, what we've essentially seen is that using this approach, we can capture all of the information that we can get at the bulk, in addition to capturing some of these features about the heterogeneity of these perturbations and conditions. So this kind of like gives us insights into the spatial associations in terms of how the cells are moving spatially. But what is happening when in time, right? We're limited in this sense because the assumption in all of, in computing these parameters for motility is that there's time invariance, right? And what that means is that we're taking time averaged values along these different time lags, right? So that we can't really extrapolate anything in real time because we're assuming overall everything's just going to average out, right? So the way that we try, there's a couple different ways that we can try to capture this temporal behaviors, right? And then, some of it is that we can, go back from the fundamental equations and add in the time component and rederive everything, right? But I think there's an interesting way that we can do it, which is we can take movies that are longer, break them up into chunks, which is like 2.5-hour chunks, as I showed before, and then stitch them together over long times, and then we can capture that temporal dynamics, right? So within each of these 2.5-hour blocks, the time invariance assumption is valid, but then we're still able to capture the behaviors kind of like temporally. So we went ahead, did the same type of analysis as I showed you before, this time just with 8 hours of movie, right? So we had these 8-hour-long movies. We computed sort of like the clustering analysis, found that there were 25 clusters. These 25 clusters are defined based on the 8 parameters the same way, break into like these 4 different modules again, and then you can see the 25 distinct sets of behaviors, right? But then the question is now, how do they map onto each other, right? So within these 8-hour movies, we can chop them up into 2.5-hour blocks and connect them. And what we see is that instead of mapping, state 1 in the 17 to state 1 in the 25, we sort of group them into the 4 modules, right? So how do the 4 modules map in one space, map to the other 4 in the other space? And what we see here is that there's these interesting dynamics in terms of the temporal nature of these cells, where SG1, which is the red, seems to be very stable, right? So a cell that's moving, not moving much, tends to stay not moving much for consecutive 2.5 hours. And cells in the yellow, which are the fast movers, right, the far movers, if they're moving a lot, they tend to move a lot. But there are certain cells characterized in SG2 and 3 that seem to be more, they feed into many, right? So they're more plastic in their phenotype and are able to transition between some of these different modes of movement more frequently. So using this temporal nature as well, how can we understand, right? So I'll show you this nice movie. These are T cells moving in 3-dimensional gels, and they're actually navigating their space, right? So you can see cells that move a lot, cells that don't move much at all, and so forth. So in these cells, we expose them to different microenvironments, right? So the microenvironment here we define as the collagen concentration, right? So we're changing the local architecture of the collagen, right? So we're increasing from 0.5 to 4 milligrams per ml. And then that is simultaneously changing pore size, fiber alignment, stiffness, and a variety of different mechanics, right? And then what we see, interestingly, is that just based purely on the trajectories, we can see that there seems to be some sort of biphasic response, right? So cells tend to move a lot at low collagen concentrations and then sort of like shift up, kind of like decrease their motility and then comes back up. So we can see that at the bulk. We can see that in terms of our classifications, in terms of the heterogeneity. There's also a biphasic nature in the heterogeneity. And then if we now map it spatially, and this is a slightly different spatial map, we can also see that we can capture this dynamics in terms of the cells moving. So you look at the lower green, we're going from blue to orange. You're traversing up and then coming back down, right? So it's always showing this biphasic nature in terms of the spatial associations, right? But then here's one of the things that I'm super excited about, is in the sense that how does that look in terms of the temporal dynamics, right? And what we also see is that we can capture that based on the temporal dynamics as well, where you can see the dominant modes are yellow for 0.5, yellow for 1, then it's starting to switch to red. It switches over to red at 3, and then comes back at 4 to yellow again, right? So this is showing kind of like we're able to understand both spatial and temporal behavioral dynamics in terms of these cells. So why is this important? One of the key questions is in the sense of how dimensionality really influences our cells, right? So there's this big debate, is it 2D better than 3D? And how do we really reconcile that? So we asked, can we take cells, put them in a three-dimensional gel, a physiologically relevant one, and then I'll put cells on a 2D surface, right? Can we map them and see what really encodes this difference in terms of the dimensionality that we can use? And we use 60 different conditions, right? So these were HD 1080, so it's a fibrosarcoma cell line. And we had 30 different perturbations, right? So we knocked down many genes that associate with the cytoskeleton and then try to just expand the universe of potential different moves. And then, we're able to see in this t-SNE space that the 2D cell conditions cluster by themselves and the 3D conditions clustered by themselves. If we calculate the spatial heterogeneity, we see that 3D seem to have higher heterogeneity than 2D, right? So there's an inherent increase in the cell-to-cell variability in terms of their behaviors. So can we use this, just the heterogeneity, to predict which cell is in 2D or 3D? So I'm just taking conditions blindly, and I'm based on that spatiotemporal pattern and the heterogeneity, can I tell you if that cell is moving in 2D or in 3D? And we can, this can be extrapolated to many different conditions. And what we're able to show is that we could predict with a 98% accuracy cells that are moving in 2D or in 3D based purely on the variability within the population, right? So to do this, we computed a couple different measures of the heterogeneity that captures both the spatial entropy as well as the temporal entropy and the conditional entropies, right? Because there's a certain probability that a cell who starts off in SG1, the red, would stay in SG1, or a cell that starts off in SG4, which is the yellow that we saw, would stay, right? So using this, we can really combine some of these different features to understand what are some of the phenotypic determinants that kind of govern some of the behaviors that we see and exhibit, right? So what I hope that I've shown you so far is that, biophysical properties of cells can offer insights in terms of aging, in terms of both how that information is encoded and how it's propagated, and that cellular heterogeneity really plays a key role in terms of this propagation. So some of the work that we're building on this is really investigating how some of these behavior patterns are changing in circulating immune cells, because circulating immune cells, as they traverse the body, likely adopt an ensemble phenotype, right? So we're trying to capture this ensemble aging phenotype and really using combinations of baseline measurements as well as perturbations, whether that be chemical perturbations, mechanical perturbations, or exposing cells to what I like to call obstacle courses, right? So you put them into these microfluidic channels that you're able to control the topography of the cells and see how they make decisions and use that into our predictions. But then a key piece of it is that we're able to understand mechanistically what's changing biophysically, right? And understand these patterns that we can use to stratify people. But the way that we can intervene potentially is based on an understanding of the molecular networks or the underlying molecular states that drive these phenotypes, right? So simultaneously, we're working on trying to incorporate molecular profiling with these biophysical profiling to really start to connect the different states that we have and understand how we can directly manipulate and modify some of this. So in the last few minutes, I'm going to go through just one super exciting project that we're just starting to get data on. So my first PhD student over the last year has done like an amazing job really putting through some of that. And I just wanted to share with you just a little bit. So we're interested in this notion of cells transform into what we call senescent cells, and those cells accumulate with age, right? So these senescent cells are very inflammatory. They induce a lot of disruption in communication, but also defects in terms of the microenvironment. But the question in the field is, when is senescence good and when is it bad, right? We know it's good in the context of wound healing, and we know it has bad associations in the context of cancer. But are there instances where it's beneficial, and how can we identify those? So we've been working on developing what I call a senescence reference map that really tries to take advantage of these biophysical properties of cells combined with biomarkers of protein-based biomarkers, and then in light of the kinetics of these associations with age, can we develop new ways or new approaches that we can streamline the use of senolytics, right? And senolytics is a new class of drug that seeks to kill senescent cells and sort of like it reverses some of the age-associated dysfunctions. So we're doing a lot of high content morphology, right? Here I'm showing you a 10 by 10 image, right? So these are 100 individual tiles of cells, and we're able to use image processing approaches to segment these cells and then also associate them with classic biomarkers, right? But the issue with the biomarkers in the field now is that none of them are perfect, right? We know that lamin B1, for instance, should go down, HMGB1 should go down, P16, P21 should go up, beta-gal should go up, but it doesn't happen the same in all cells. So we've been multiplexing this in imaging. So my grad student have been staining all these different biomarkers within each of these cells and then imaging them to quantify morphology and these biomarker status together. And again, we've been using some of these, single cell type approaches where we can map the morphology, use just the morphologies to map in multidimensional space these different conditions, right? So we induce senescence in 4 different ways, and we're seeing that we're moving from one side to the next of the UMAP, right? All the controls seem to be on one side, and all the induced seem to be on the other side. And we're seeing these interesting effects in terms of age, right? So if you look at an older 85-year-old versus a 23-year-old, you see that the pattern is very different in the sense of how where the control cells are located and how they're shifting over time. We could then layer in these different protein biomarker information purely on a morphological state and still see a pattern in terms of the protein expression of these senescent markers in a morphologically defined space, right? So what that tells us is that the morphologies of these cells are capturing some of the underlying molecular states of the cells. And here, this is just showing P21 expression, and we're seeing as a function of these inducers, we're shifting the population towards these, senescent-like. We can do this across multiple different biomarkers. And interestingly, we have biomarkers that go up with senescence and go down with senescence. So we can start to capture these intervening populations and so forth. And then I'll just jump through. We're able to show morphologically they're different based on this clustering analysis. And then we can see that it's actually biasing. And then there's this association in terms of we could define regimes of senescence where we can start to map the transition or the kinetics of cells being induced into senescence. So we could identify these subtypes of senescent cells, and inside that, cells that are more prone to senescence, cells that are resistant to senescence, and understand these dynamics that I think is going to really help us. So we're working a lot on the kinetics right now, and in terms of the microstates, in terms of state transition analysis. But then we're also collaborating with Denny Wirtz and Ashley Kemian and Sashank Reddy, who's a plastic surgeon, to reconstruct this in actual human skin to understand this physiological context of senescence. And with that, I'll stop. And I'd like to thank all my students who contributed to this work. Most of it was done by Dr. Debanil Maiti, who's my postdoc, and first-year graduate student Pratik Kamat. Thanks.
Dr. Phillip, thank you for the wonderful presentation. Before we get into the Q&A, I just want to call everyone's attention to our next hybrid seminar, which will take place in 3 weeks on August 1st. We have a little bit of a break because of spring break and whatnot. On April 1st, I'm sorry, I said August, didn't I? On April 1st, I think everyone knew that, we will be hearing from Dr. Zhu Han from BU as she presents voltage and calcium imaging of biological neural networks across spatial and temporal scales. We are also excited to announce that on April 23rd and 24th, Columbia Bioengineering is hosting the 48th Annual Northeast Bioengineering Conference here on our Morningside campus. And the abstracts for that are due on March 21st, and registration is now open. you can visit the website nebec.org for more info. And please follow us on social media to get all of the latest updates. You can see our social media information right here. Now we're going to switch to the question and answer session. We have a bunch of questions online. We also have some questions here in the audience, I would imagine. One of the questions that came in online, Jude, was skin has different layers and they are chronologically different. How do you determine that cells collected are chronologically similar from individuals with biological differences across the age ranges from 2 to 96 years to make that comparison?
Right. So we focus only on dermal cells, so cells within the dermis, and we took just dermal fibroblasts. So that's one way we just looking at one population. But in some of the work that we're starting now, where we're trying to do it in reconstructed skin, we will be able to find associations between dermis, epidermis, and the different stratas of the skin.
Cool.
Hi, thank you for the presentation today. It's really a great tool to specifically make these phenotypic clusters and especially finding phenotypic variation. So you hinted at it in your last bullet on future work, but it would be really powerful if you could somehow kind of take these phenotypic clusters and then find a way to either put them into like single cell sequencing or something, and then mark them directly back to phenotype to their like genotype per se, and then you can like there's just a lot you can do with that in terms of like the aging cells, healthy cells, even tumor cells. Do you think that that's something that could realistically be achieved through microfluidics, separating out the different phenotypes or tagging them with fluorescent marker or something? And do you think it would be helpful in the long term?
Yeah, definitely. You're spot on, and that's some of the things that we're definitely thinking about. I think it is possible. There's a couple different ways that we're trying and thinking about getting it to work. In terms of just purely incorporating into like present kind of like pipelines for single cell may be difficult just because of the dynamics. What we're thinking about doing is in some cases doing like large CRISPR screens, for instance, where you can manipulate and then you're able to assess the phenotype in the wells, kind of like Dr. Jose does. And another way we're trying to do is that if we could do single cell sequencing, understand the different probes, we can do something like fish, right? Because we're image based. So we can do just like fish dynamics and then map the population. Yeah.
Thank you.
You're welcome.
Questions? One question. Can you hear me? One question from Allison McGugan online. Allison wrote, "It's a really wonderful talk and it was great work. Have you been able to assess how changes in cellular heterogeneity due to microenvironmental changes such as 2D to 3D or plus or minus cytokine compare in magnitude to changes due to aging? So will particular environments overpower signatures of aging, and are there microenvironments where you're most likely to be able to detect age-related signatures?"
Right, and so the short answer is we're not sure yet. We're working on that. So we're doing a lot in terms of inflammation. So this inflamed phenotype that is characteristic of older individuals really informs or could enable or maybe suppress some of the intrinsic aging phenotypes. So we're actually measuring that. So short answer, we don't know exactly yet the magnitude of the differences, but we know that they sort of combine in terms of giving us things that we can measure in terms of age.
Another question online from Mary Reiger. How might you incorporate spatiotemporal patterns of cues in addition to spatiotemporal patterns of behaviors / responses?
Yeah, that's an interesting one. And I think some of it could do with like intravitreal microscopy, right? So one of the studies that we just started on is looking at the dynamics of T cells and B cells within the germinal center, right? So the germinal center is this like well spatially defined that has like very interesting like characteristic cues, right? Because when we experience this antigen, these structures are formed in our secondary lymph nodes where the B cells have to be trained to produce the right antibody, right? So within the structure, it's very spatially defined, and there's really a tight spatial cue, right? And we're looking at these behaviors within these different spatial cues. So that maybe could help us understand some of those spatial associations. But in terms of in vitro, there are ways that we can develop with microfluidic based channels or hydrogels that you can sort of like tune spatially the cues and the responses.
Another question online from Pule Ravini. How do you deal with aging differences due to plastic adherence? Is there a difference in how H2 versus H96 cells respond to the plastic in terms of aging in vitro? Is there a feature you can monitor because this could be different from in vivo aging of the fibroblasts?
Right, that's true. So what the logic that we have in terms of like taking these cells completely out of physiological context and measuring is the fact that the intrinsic differences that are age associated or maybe heavily age associated would inform all of that, right? So even if they're adhering less, that is something that would come out in terms of our morphology-based measurements that would still inform. It's not telling us about the intrinsic biology because at that point, we're not studying the intrinsic biology. We're just trying to capture the age-associated difference that we see.
And there's one final question online. This is a nice question to end with. It's a little bigger picture. Which companies are leading the way in this field? I wonder if this is from someone who's looking for a job, perhaps.
So in terms of the aging space, two companies come to mind. They do a lot of the classic work like Calico in California and the new Altos Labs, I think, which was announced. I saw it on Twitter a couple of weeks ago. That they're really trying to like reprogram aging. So I think like those are two like pioneering like very heavy companies in that space, both in California, I think. But then we're hoping to eventually kind of like based on what we find, we may be able to start something in a couple of years, right? We always have our eyes tuned to sort of like entrepreneurial endeavors.
Sounds exciting. All right. Cool. All right. Well, thank you very much for this great talk. This weekend.